That applause, I haven't even said anything. So what I want to do today is tell you a little bit about who I am and how what I've done in the past largely guides what I'm doing now. I also want to tell you a little bit about the story that I've had and how I got through the years you guys are in now, some of the stupid or crazy things that I did then, but how those things actually shaped what I became and shaped my decision-making process that led me down a path that ultimately lets me do this thing that I'm doing now. So I get to teach at a university level, which is really what I love doing. I've had the opportunity to do some other things. This is the greatest thing. So that's what we're going to talk about today. Really the what, the why, and the how of Dr. Trott and his lab and his students and what goes on, in the genetics classroom. Many of you I have had as students, so if you're not freshmen, I've had you probably in genetics already. Some of you have been my Southern Connection students, and so that's been fun. The rest of you, GB2 students, I get to get you next. So we'll see you all in the fall. That'll be a good time in genetics. That's the major class that I get to teach here. I'm not really a geneticist. Shh, don't tell anybody. Actually a cell and molecular biologist, but we'll get to that in a minute. So, my life has not always been this perfect straight thing. There's been a lot of ups and downs, certainly during my college years, trying to figure out what on earth it was I was going to do. And that's really what I want to really start with. How did I get down this path with twists and turns and undecisions and corners I couldn't see around to ultimately get to the point where, all right, I have a job I really, really love. I feel really blessed by God. It's been a good thing, and I get to be with you guys every day. As Ronald mentioned, I think I have a beautiful family. It's a good thing they look like their mother. So, but they're really, really important to me. And this idea of family and time as a unit and kid time and wife time has been something that's actually driven my decision making ever since I was in college. It's been an important thing for me that I can have a family life. I grew up in what I consider a pretty healthy family, and I wanted to try to replicate that as a professional, as an adult. Lord willing, pray for me that we can continue on with that. So, I went through high school and survived, praise the Lord, and started off in college, and it was really no decision for me. I was always going to be a biology major. I was always going into science. That was obvious. I was just fascinated by living things. I was the kid who watched Wild America as a kid. You don't know what that was because you weren't born then, but it was a really cool show with Marty Stauffer. You could watch it on YouTube maybe. Who loved science, who's always outside playing with the bugs and the frogs and everything. I caught every single frog in the pond every summer, more than once. And that was just me. I loved it. And so going to biology major, that was easy. The decision started to come after that of what on earth was I going to do with it? And we'll get to that in a second. So I was in college. I was having a grand time. I knew nothing. I was just studying away, doing stuff, didn't know what I was doing, had no idea where I was going. In fact, I did a lot of this. Played a lot of basketball. Do you see me on there? I look just the same today, so it's pretty easy. That dorky guy right there. Played a lot of basketball. Was okay. I wasn't great. Still not great, but I still like to play. In fact, I made the All-American Scholar Athletes list one year. This is a national award. This was pretty cool. Look, this is me over here. This is my name right there. See, Tim Trott right there. That's pretty good, right? Still play basketball. Ha! So that was actually part of my life, was playing basketball. I studied all of my college career in a bus, playing basketball. I still don't know how I did that. I didn't sleep. It was awful. Don't do that. Especially those of you pre-meds, don't do that. So I got into college and I was like, okay, we're going to be a biologist. And that seemed like an obvious thing coming out of high school. I had no idea what that meant. So I started off, I actually was a biochemistry major. That was a bad idea. So I started my freshman year. I took GB, I took GChem, I took Calc 1, first semester. I was a 4.0 student in high school. My first semester, I got a B in GChem and a C in Calc. Tearing my heart out, right? You guys been there? So I said, well, I don't have to worry about that 4.0 thing anymore. That's kind of liberating. Took Calc 2 my second semester, said, this is ridiculous. I don't like this. I really don't want to be a biochemistry major. What was I thinking? I don't really want to be a biochemist. I just thought it sounded cool, biology, biochemistry, that sounds cool. Let's do both. Dumb idea. So then I just switched back to biology major and thought that'll be fine. So I finished up my freshman year, just a biology major, and had no idea what I was going to do with that, just biology, because I like life, and I'm playing a lot of basketball, and I'm not doing anything else, really. I didn't have a girlfriend. I don't know what I was doing. And then end of my freshman year into my sophomore year, I said, I'll be a pre-med. That's what all biology majors do. I'll be pre-med. That sounds good. So I'll be pre-med. So we continued on in my sophomore year, pre-med, got about 3/4 of the way through my sophomore year, spent a lot of time talking to doctors, looking at family friends who were physicians, and just that wasn't for me. It became very, very clear that I really don't want to be a physician. And part of it stems back to that family life thing, I see the family life that my physician friends had, and that was just like, I really want to see my kids occasionally. That would be nice. And so really didn't want to be a physician. I think I could have been a fairly good physician, honestly, and I think I would have enjoyed it, but just no, not for me. Let's do something else. So I ditched the pre-med thing. But then what? Now what do you do? You're a bio major, you're not pre-med. A dentist? No, forget that. Right? So what are we going to do? So I thought, well, my mom's a teacher. My dad was a professor, he was in religion, something else. My brother was kind of thinking about teaching. He was like, well, let's do teaching. That'd be fun. I'll teach high school. I could enjoy that. So I started off in secondary education. So I was essentially another major. So I was biology, secondary education. Took all education classes, how to be a teacher, pedagogy, like making lesson plans and everything. During my junior year, I actually got to teach at the local high school. It was very similar to here. We had Adventist college and a little, the Adventist school right next door, and I got to teach there because they lost their science teacher. So I taught all the bio labs for high school for a whole year, just me. The professor didn't come. I was sort of under one of the college professors. Just me. It was actually really good. Learned a lot, got to develop lesson plans and teach. But you know what that 1 year did? Cured me of wanting to be an education major. I loved the teaching part, but high school students are possessed. They are insane. Maybe one of them actually wanted to be in class, and you're doing all this song and dance trying to teach something, and it was, the teaching was fun, but the not being interested at all thing, I have a high school student now, pray for her. So I actually finished the entire secondary education major. I did my pre-practicum hours. For those of you who know what that means, I spent weeks in a high school observing and helping, and I dropped it short of my practicum, didn't do it, walked away from it, and didn't finish it. So my degree doesn't say anything about education. And so I went into my advisor's office just almost in tears, like, I have no idea what I'm going to do now. I don't want to teach high school. I don't want to be pre-med. I want to be a biologist, but that doesn't, I don't know what that means. So what am I going to do? Well, he gave me some advice that changed my life that day. He came in and he said, you should go to graduate school. I know you'd be a great teacher. You are a great teacher. So why don't you go to graduate school and teach college? Go to graduate school. 6 years later, you come back and take my job. He wanted to retire. I said, I don't know what that means. How do I go to graduate school? I would go get my master's and then go get my PhD. What does that mean? He said, no, you just forget the master's. Just go get your PhD. I can do that? Yeah, to go do that. Okay, well, how do I afford it? I don't have any money. My parents work for the church. We're poor. He's like, no, graduate school is free in the sciences. They pay you to go to school. I was like, what? Why didn't you tell me this? This is like the greatest deal ever. So that was it. After that point, I said, I'm going to go to graduate school. I'll go get my PhD in biology and try to become a college professor. I think that sounds great. That was really neat. And that's actually what I ended up doing, in fact. The light kind of came on, right? I was like, aha, that sounds really good. Let's go to graduate school. So I spent my whole senior year working on going to graduate school in biology, applying to schools, and eventually got accepted into the University of Vermont for their cell and molecular biology program. That worked out really, really well, too, because my two favorite classes in undergrad experience were cell and molecular biology. I know you think I'm crazy because you hear horror stories about how hard it is here. That's because it's awesome. Okay? And botany, oddly enough. Who likes botany? What do you want to study plants for? Those are two things that just really kind of clicked with me. I just thought plants were awesome, and I really like cell biology. They seem opposite ends of the spectrum, and they are, but they were cool for me. So I got into a program that allowed me to do both, and that was really, really great. I got to combine the two greatest things in my life, and I thought, oh, this is great. I'll get to teach things like genetics and cell molecular biology and developmental biology and microbiology, and I really like those subjects. I get to teach them to students like you who actually might want to learn. Maybe. Right? A little less possessed than high school students, right? You guys are awesome. So that was really the journey how I went through college. And, lots of twists and turns, lots of times when I had no idea. I look back on those years and I'm like, Lord, thank you for keeping me safe and sane and helping me make wise decisions that led me to a path where I can do something to glorify your name and really enjoy what I'm doing. The other thing that happened to me in college was I got married. This is me, my senior year of college, and my wife, Selena. This was our engagement photo. I was 20. I got married when I was 21. So I graduated from college, got married in July, started graduate school in September. Right? No gap year, just go on. Greatest decision I ever made was that one right there, by the way. School, never mind. We've been married 22 years. It's been a good thing. Yahoo! So we both moved to Vermont. I enrolled in the Cell and Molecular Biology program at the University of Vermont, where I got to study plants and use plants as a study system to ask questions about how cells work and how genetics work and how plants use the proteins that they make to drive the functions that they have. And I want to tell you a little bit about some of that work and how that work has really sort of guided the stuff that we do here. And it's been a lot of fun. I hope that many of you go on to graduate school. It's a great thing. I hope that maybe someday one of you comes back and takes my job. Not yet. I'm not done yet. But, give me 20 years or something maybe, and then we'll see. So the lab that I entered was a lab. It was actually in the botany department, but we were doing cell and molecular biology. It was kind of a neat conversion. I worked in the botany department with a bunch of plant biologists. Actually, the guy downstairs was a photosynthesis guy, and he actually wrote the chapter in the Plant Phys book on photosynthesis. That was pretty cool to actually talk to the guy who knew about photosynthesis. Well, the group I was in, we studied cell wall proteins. So this is a cross-section of a herbaceous plant. And you're basically just looking at anything that's left there are cell walls. The idea is that the different colors represent differently stained cells that have different components and different thicknesses of the walls. The walls are not uniform, and the idea is that different walls have different functions. And so the main question that we asked in our lab was how do differences in those cells, and particularly in the cell walls, drive functional differences of types of plants? They live in these little boxes. The boxes are constructed differently. They have to be doing something with function. Let's figure out ways to ask and answer those sorts of questions. So the particular proteins that I worked in were in a plant called Arabidopsis thaliana. It's essentially a lab rat of plants. It's a weed. It grows everywhere in the world. As a weed, you step on it all the time and don't think about it. Shame on you. This is an electron micrograph of the surface of an Arabidopsis leaf. This was some stuff that we did while I was there. It was a lot of fun to do all kinds of cool microscopy. But you see sort of the puzzle-shaped pieces there. The puzzle-shaped pieces are the epidermal cells. And then the little mouths that you see up there are stomatal openings. The stomatal openings, you guys studied plants yet? You talk about plants? Did you talk about stomata? Okay, so stomata are made up of two cells. What are the cells that surround stomata called? Guard cells. All right, so they're like these little kidney bean-shaped cells, and through changes in turgor pressure inside those cells and osmotic pressure, they can actually open and close and regulate gas exchange and transpiration from the inside of the leaf to the outside, which is really critical in driving plant physiology, water movement throughout the plant, photosynthetic rate, all those things really impinges upon stomatal movements. It turns out the proteins that we studied in our lab were found in the cell walls of guard cells, and that made them particularly interesting because those are interestingly functional cells. They have a really unique and particular function, and the walls of those cells are constructed in a very sort of different way. I want to just sort of show you a picture up here. So this is the stomatal pore here. This is one guard cell on this side. Here's another guard cell on this side. And the arrows on the one on the left point to these kind of lipped or shelf-shaped structures that surround the pores, and that's all cell wall material. It's all this interesting thickening that the end of the story actually is that it actually acts like a spring so that when the turgor pressure inside guard cells relaxes, it pulls the guard cell closed. It's actually kind of neat. And then the guard cells pump themselves up with water and they actually push against that spring and open back up. And if you remove some of that cell wall material, the guard cells won't function properly. So we ended up having a protein that was expressed in those cells. I want to tell you how we got to that point and figured that out. So the first thing we had to do is we had this protein, we had this gene, and we didn't know where it was expressed. So we had to develop a tool that helped us understand where in this plant this gene was expressed. It wasn't everywhere. It was only in certain places, but we had to figure that out. How do we do that? So we developed this gene expression tool, and what we did there is we took the GFP reporter gene. You guys talked about GFP at all in general biology? We will in genetics. So those of you who've been in genetics are like, oh yeah, I got this. I know what that is. So this is a gene from jellyfish called green fluorescent protein. and it fluoresces green under ultraviolet light, which is kind of cool. So we use it as a reporter gene to tell us when and where the gene is active. I'll show you how this works in a second. The way we did this was we took the regulatory regions, or the promoter, from our gene of interest. It was called ATPRP4. It's just a name, doesn't matter, right? But that was our gene of interest. We took the DNA that was upstream of that gene, the one that drives transcriptional rate of that gene, called the promoter. We copied that out, made this genetic construct where we tied that to our GFP gene. Oh, wow, here we go. Tied that to our GFP gene. We built that back up in bacteria. We transformed it back into plants. So we made a transgenic plant where we've genetically engineered it. It's actually remarkably easy to do in Arabidopsis. So we basically added a new gene in. And what that would do for us is anywhere our normal gene, ATPRP4, is transcribed and translated, this gene would also be transcribed and translated because it's using the same regulatory sequence. So it reports to us where our normal gene is active. So using that kind of thing, we transform it into plants, and then we just look at the transformed plants under ultraviolet light and say, where are they green? Right? And we can answer the question of where our gene is active. So this is fluorescent microscope images of plants that have been transformed with this sort of thing. And I want to show you two places where it's transformed. This is a stem up there, and A is a stem, and B is the surface of a leaf. And what do those look like? They're guard cells, stomata, right? So very strongly expressed in guard cells of stomata. Cool. Anybody have any idea what the bottom ones are? I don't know. It's actually a root. So turns out that it's really strongly expressed in an inner root layer called the pericycle. You guys study root tissues at all? So this inner layer is called the pericycle, and whenever there is a branch root about to form, we see really strong expression in the cell layer that divides off the pericycle. So it's really important in the initiation of lateral roots, wherever branch roots come off the main root. And those are two really interesting cell types because they have weird functions, very unique to a plant. You have guard cells that have this weird function that's different from the epidermis, and lateral root formation is a really sort of odd event. It's like growing another arm, right? That's hard developmentally. We have to do something weird. So that was cool. So we knew when and where the gene was expressed. What time is it? Oh, good time. And then we wanted to know, where did the protein go? So just knowing that the gene is on in that cell type doesn't tell us anything about what the protein's doing. It just tells us the protein is transcribed and translated in that cell. So the next question was, where's the protein put in that cell? We think that it's going to be a cell wall protein based upon some understanding of sequences and how other proteins that go into cell walls look. But we're not sure if it goes into the whole cell wall of that protein or if it only goes into particular parts. Is it just going to one side? Is it only where there's a particular adhesion junctions? I don't know. We didn't know. So we needed a new tool. So we created another genetic tool. Here again, we used the regulatory sequences over on the left from our gene normally. So we took the promoter region from that. And this time we built a whole new protein coding gene. We essentially took the whole coding region of our normal gene called ATPRP4. It had two domains. It had this N-terminal domain, which was kind of odd, and then it had a C-terminal domain on the right, which was called the PRP domain. It had a lot of proline in it. It was called a proline repetitive region in this particular particular protein. What we did is we modified the coding region by putting in an extra little bit called an epitope tag. The epitope tag was a series of about 9 amino acids that we inserted into the coding sequence. So it was about, how many base pairs would that be? If we put in 9 amino acids, how many base pairs did we put in? How many codons would we need for 9 amino acids? 9. How many base pairs are there per codon? 3. What's 9 times 3? Oh, we got that one, 27. Okay. All right, so we had to put about 27 nucleotides in there. We kept the whole sequence in frame. We didn't change anything else. And what that did is it gave us a little molecular handle. The reason we put that in there is because we could buy an antibody that would bind to that epitope tag, and it would allow us to find this protein anywhere in the plant. Okay? So you put in this little epitope that you can buy an antibody against, then you put it back into the plant. Wherever that gene's expressed, it makes this modified protein that should behave about like the normal protein does, because we only changed it a little bit. And we could use this commercially available antibody and find it anywhere in the plant and say, where in the cell wall is it? Is it everywhere, or is it in particular places? So doing that kind of stuff, we did some really fancy microscopy. This is called confocal microscopy. We also did some, I can't remember the name of the other one. That wasn't the name. It was a deconstruction microscope where we did optical. It was called a deconstruction microscope, but you actually optically reconstructed things, but that was just the way it was called. So what you're looking at here is, again, remember our picture here on the bottom right. This is what 2 guard cells look like, and I've sort of false colored this. We actually used this technique to look at 2 different proteins that were related. One of them was called PRP2, one of them was called PRP4, but that doesn't matter. But they're very similar proteins that are expressed in these same cells. Turns out that one of them was only expressed just in these lips or the shelves around there. That's the only place we could find that protein in the cell wall. We thought, oh, that's so cool. It's only in one place. It has to have a really neat function because it's just there. The cell's doing something unique with it, right? Unique localization almost always means unique function. The other one was even cooler, I think. It was only expressed on the inner surface of the stomatal pore. It wasn't anywhere on the outside. So these other 3 pictures are actually optical reconstructions of where that protein's localizing on the guard cell walls. And essentially what you're seeing here, right, here and here, is this just lining in here lit up with an antibody that's tagged with a fluorescent marker, right? Is that making any sense to you? You get this neat torus structure, which is essentially just the tube of the stomata. And we just thought, oh, that's so neat. We got these 2 proteins that are these really cool localization. And so now we know when and where they're expressed, and we know where the protein goes. Do we know what the protein does? No, right? We have no idea of function. We can guess. It's probably important to stomatal function, but because that's where it is. But we have no idea what the protein does. So how do you figure out what protein function is? What would you do? What's the next step? I don't know. We weren't really sure either. No, we knew. So the next step is really to create a plant that lacks that protein. So geneticists are really, really good at breaking things, right? So we take a protein, we want to know what it does, we break it or delete it, right? We do something that prevents its normal expression, and then you look at the organism and say, is there a phenotype associated with its loss? And so that's what we did. We went searching for some mutants that did not make this protein. We used a process called insertional inactivation, where we identified 3 different mutants that had a really large piece of DNA inserted into our gene of interest or very near our gene of interest. So you can see here's 3 different lines. Don't worry about it. But essentially, the tDNA is a great big piece of DNA that was inserted. It's huge. It makes a frame shift mutation, so the protein never gets translated in a proper way. So we were identified plants that did not make this protein. It wasn't necessary for viability, but we could look at them. So we actually had to confirm that they weren't making the protein. We did that in a couple ways. This top panel here is a process called reverse RT-PCR, reverse transcriptase PCR, sorry. And what it does, it looks at mRNA levels. And so the first one here is basically signal from a wild-type plant. We're able to detect the mRNA for this gene. In our mutant line, there's absolutely no signal, meaning there's no mRNA made for that gene. So we're saying, okay, they're not transcribing it, they're not producing an mRNA, that's good. Let's also confirm that they're not making the protein. And so we used a different antibody, which actually recognized our wild-type protein. We had this one later. And we could show in a wild-type cell, we can identify that protein in the lips of stomata, whereas in the mutant one, which is this one here in the middle, there's absolutely no signal, we're not making protein. Okay, so we have this mutant line, not transcribing or translating our protein. Is there a phenotype? And the answer was yes, there were some weird phenotypes. This is what the plant looks like normally. Wild type is up there on the right. It grows with this little rosette of leaves, and then once it gets mature and has enough leaves, it sends up a reproductive bolt and flowers, these little tiny, pretty white flowers with yellow stamens in them, microscopic things. So the plants on the left, that's normal. Plants on the right, normal. Plants on the left are mutant. Do they look different? What looks different about them? They're smaller, right? And they're flowering. These plants are the same age. So the mutant plants are actually flowering earlier. If you're a plant biologist, that means stress response. So the plants on the left, the mutant ones, are stressed. They're trying to reproduce before they die. Okay, so we had a stress response in these plants. They were surviving, but they weren't happy. Their leaves were smaller, so you can see down here, leaves are smaller. Their seedlings had some odd phenotypes too, but it still didn't tell us what the protein was doing. It just said, well, it's important for proper function of stomata, probably. So we did some careful work on stomatal conductance, which I'm not going to show you right now, but we could actually show that the stomata weren't working right, and that was the key feature for all of us. was that we could show that even though there was the same number of stomata on wild type and mutant plants, they weren't opening and closing at the right times. And so the plants weren't able to transpire well, and they ended up with a bunch of other odd things happening. So we could quantitate rosette leaves and rosette diameter. And then the last piece of this story, and I'm going to turn my work over to these guys, is the keystone feature for any sort of genetic process like this. You understand where a gene is expressed, you find where the protein is, you identify a mutant phenotype, and then to confirm that, in fact, those mutant phenotypes are probably the result of the mutation you made, you fix it. And that was the last step that we did. And so we took these wild-type plants, right? Had a wild-type plant with some sort of phenotype on the left. We could delete our gene and produce some mutant phenotypes, and this is just one of those mutant phenotypes that we identified. And then if we added back that epitope tagged version of the protein that we made at the beginning, so it didn't make any normal protein, but it made our epitope tagged version, all the mutant phenotypes went away, right? And so it's a nice way to sort of close the loop. In fact, all of those phenotypes that we observed are a result of our mutation. So we learned a lot about how that protein is expressed and made and translated and where it goes in the cell and how it affects protein function, how it affects overall cell function. That make a little sense? Really fast primer on forward genetics. Okay, so that's driven a lot of the work I've done here. Real quickly, and these guys have a few minutes left. What time is it? What time are we over? Oh, we got tons of time. Sweet. I was rushing to get through with these guys. Okay, so what this work has done is largely driven the research work that we've done here. Being a cell biologist and still appreciating plants, I've tried to focus most of the work we do here with student projects and undergraduate projects along those same lines. And you guys have a really huge blessing here at Southern that you have a biology department that has space and equipment and funds and professors who are interested in doing this, which is actually fairly unique at an undergraduate institution to have the ability to do research. Any of you guys who are really interested in doing research will be able to do it. If you come and talk to us and you really want to do it, if you just kind of come on a whim, I don't know, I might want to do research, we'll be like, we might not want to work with you. But if you come and are really interested and we can tell that you're motivated about this, we'd love to figure out a way to put you in. So we've done a bunch of plant projects over the years. I've actually done some continuing work with several different groups looking at some of these proline-rich protein genes in a number of different organisms, a number of different types of plants. We've looked at sort of local flowering species here. We've identified them in a number of different plants. We've done some surveys on them. We've done some things about how to identify the genes in different places. So these are some groups that have done this over the years. You guys probably don't know these guys. Any of you guys know any of these guys? This is beyond your time. We'll get a little closer. You might recognize some of these folks. Yeah. So we've recently started a group looking at berberine expression in golden seal and the effects that berberine have on plant growth and antimicrobial effects. And so that's work that the group on the left did. We were trying to learn how to grow golden seal in our growth chambers, and we tried a bunch of different conditions. That was a lot of fun. Shayna continued that. Shayna is actually at Kettering now doing a PA, so that's kind of fun. Mackenzie just graduated a couple years ago. She's at Loma Linda now in medical school. She did a project doing DNA barcoding in oak trees locally. So we have, and I'm going to give away a little bit of your presentation, just a tiny bit, it's okay. We have a bunch of oak species here in Tennessee. You guys ever walk the biology trails? You should. No, I'm a biologist. Why would I go outside? I want to do that. I want to go to med school. I don't want to go outside. So there's a lot of neat oak species up on the biology trails, and you can identify white oak and red oak and pin oak and blah, blah oak and other oaks and other oaks. And then there are species, there are individuals up there that you look at them and they're not this, they're not white oak, they're not pin oak, they're not red oak, they're not southern red oak. What are they? They're actually oak hybrids, right? So they're actually a hybrid of two other species, and they're difficult to key out because they have features of both. So we had this neat idea that maybe we can use something called DNA barcoding to essentially look at parental heritage of these trees and say, okay, white oaks have this sort of DNA barcode, red oaks have this sort of DNA barcode, and these hybrids in the middle, we could figure out who their parents were. And so she was trying to set the groundwork for that project, did a lot of neat work with us. And Devin's going to tell you about some continuation of that work. Luke is here. Where's Luke? Is Luke even here? No, Luke's at a caving meeting. Luke is doing some continued work with golden seal with me now. He's actually in the process of writing his thesis. He did a really neat project looking at how berberine, which is one of the secondary alkaloids of the plant golden seal, actually has antimicrobial properties. And he did some really neat microbiology stuff and then tied that with some HPLC work, and it has a really neat story to tell. So you'll probably hear some of that eventually. All right. What's next? This. Okay. So now, what I've done is I brought some of my current students with me, and they're going to talk to you a little bit about projects that they're currently doing, and all of their projects involve plant work. I try to get my students to stick to plants. It doesn't always work, but we mostly do. So I have Devin Cummings and Sierra Garrett and Claire Brewer. Do you guys know any of these guys? All right, so they're going to talk to you a little bit about their projects and how that goes. So Devin's going to be first. We'll give Devin some applause, too.
Thank you. Thank you, everyone. All right. I think some of you guys are in the bio class that I tutor for. I think I see some of you guys, Thanks for coming out. Yeah. Let's see. Okay. So when I asked Dr. Trott to do a project with him, I was thinking of more of a genetics project because personally, I don't really like plants. So, yeah. So I tried to get the closest thing to a genetics project as I could, and I decided to take up Mackenzie's project. As Dr. Trott said, her project was actually looking at DNA barcoding. And so as he kind of implied, DNA barcoding is basically getting the sequence of the DNA and comparing it to something on an online database, right? And so what she would do is she would collect leaves, Mackenzie would collect leaves and grind them up and extract the DNA from them, then send them off to this plant somewhere out in Texas, somewhere, California, I don't know. And then she would get the DNA back, the DNA sequence back, and compare it to the online database to see whether or not it matched something that was already there. She mostly worked with northern red oaks, that tree in the bottom left, and then the other tree, I believe, is a northern green? Not sure. All right. So with technical difficulties, just bear with me. All right, so why am I continuing her project? Basically, when she was going through her project, she found that a lot of her PCR results and a lot of her DNA sequencing results didn't go through. And so what I'm trying to do is I'm trying to troubleshoot that. And I am working with different DNA extraction kits to see whether or not the type of kit and what it accounts for affects the DNA yields. So basically what I'm looking at is I'm trying to see what happens if we affect the polyphenol concentration, the polysaccharide concentrations, the protein concentrations, seeing how changing those affects the DNA samples that I get. And so basically what I do in lab, I go in, put some music in my ears, and then I listen to Hall & Oates. You guys know who Hall & Oates are? Yeah. So that's what I listen to, and while I'm doing that, all right, I'll get my plants out of the fridge, grind them up under liquid nitrogen, then put them in a microcentrifuge tube and add different solutions to that, go through the whole process, then that'll take me about a few hours. Then I'll do PCR, and then the results that I get from that, I'll send them off to that plant somewhere off in the rest of the United States. And after I get them back, I'll examine the results that I get. And so this is kind of what I'm hoping for, what I've hoped for. Really clear PCR results that you can see there on the right. There's the bands are really clear, they're really intense, and they're about where they're supposed to be, so close to the 1400 base pair marker. And also I'm looking for these things called, I had it so that I would zoom in there, but yeah. So this graph right here in the bottom left is called an electropherogram. And basically what this is telling me how much should I trust the DNA sequence that I've gotten back. And so I was looking for peaks on this graph that were really clear, and I was looking for a really good sequence coming out. So I could see the A, G, T, C, and make sure that everything is okay and in order. And so this is kind of what I've been getting in reality. So I've been getting really good PCR results, really clear bands, right lengths. However, my electropherograms have something to be desired. So just like Mackenzie's project, I feel like there's still contamination in somewhere in the process. So right now I'm just working through that, trying to see what I can do to make the process cleaner, whether or not the kits are what is actually causing this, and I'm just troubleshooting. So last thing is like future work. So the project isn't complete, obviously, you know. My results kind of suck. So I'm looking for people to continue this later on. So some of the things you guys can do, you can work on the PCR conditions, you can change the heating, you can like super nuke the DNA, you know, make it super hot, make it super cold, you can change the heating profile, change the buffers used. You could look at, so there's this thing that I've been doing. This is like an original thing from my research is spectrophotometry. Basically, I'm looking at what the extract, the DNA extract that I get, what's contained in there, and seeing, so I'm shining light, like I'm pointing lasers into the fluid and seeing like, okay, what comes out, what's there. And so basically you can like use that, you can experiment with that, see like what's the concentration of different molecules in the fluid and how those would affect the later downstream DNA sequencing or PCR or anything like that. Finally, this is one thing that I actually came across a lot when I was coming up with my research project. So online, you know, you can find anything online. And basically, like a lot of the things I found were like these original protocols that people were making. They came up with their own way to extract DNA from different species. So if you want, you guys can go and look it up online, Google Scholar, JSTOR. No, JSTOR is English. Something else. Library database, McKee. Just go on, look up your own protocol and try and see if you can get better DNA extracts than I have. I dare you to do better than I have. Okay? All right. So that's my project.
Hi, I'm Sierra. And for the past couple of semesters, I've been working with Dr. Trott, and we've been studying the phytoremediative effects of Coriandrum sativum in lead-contaminated soil. So basically, what we've been asking is, can Coriandrum sativum, which is the scientific name for cilantro, be used as a phytoremediator to significantly decrease the lead concentration in the environment in which it is grown? So basically, we're growing a bunch of cilantro and trying to determine whether or not it absorbs the lead from the soil in order to be able to clean the environment, which is what phytoremediation is. So you're probably wondering, why is this even important? Well, heavy metals such as lead do not biodegrade, and therefore they negatively affect the environment which they are in, and they can affect organisms. For example, lead can negatively affect plant growth, soil microflora, and other organisms. And lead contaminations can come from everyday products, such as car exhaust and dust and paint, and things we come in contact with like all the time, but we don't really think about it. And in excess, they have really negative consequences, such as creating oxidative stress by generating free radicals and depleting antioxidant levels in organisms. And eventually, they can even lead to chronic disease and brain damage. So as you can see, if you have too much lead or other heavy metals in the environment, it's a really bad thing, right? So luckily, there's remediation, which can help decrease the concentration of heavy metals in this kind of setting. There's two types of remediation. There's physiochemical remediation and biological remediation. Physiochemical remediation includes things such as incineration, vaporization, and solvent washing, but unfortunately a lot of these are very expensive and they often leave unwanted side effects. But luckily, biological remediation is another good option and includes things such as phytoremediation, which is like I mentioned a minute ago, using plants to absorb heavy metals from the environment and clean it up that way. So the reason we decided to work with cilantro as opposed to any other plant is because there have been previous studies on cilantro showing that it has chelative properties in the body when you eat it. So cilantro is good for you because when you eat it, it'll chelate the toxins and heavy metals in your body, and it can clean you out. But we wanted to see if it'll do the same thing in the environment, like if it'll absorb enough lead from the soil to be an effective phytoremediator. So I'm sorry to disappoint you, because I know this is probably like the most exciting thing you've heard all day, but I'm not actually going to go into depth with my experiment today, because I will be coming back and talking about it more another EOG. But I will at least tell you my hypothesis, which is that there will be a positive correlation between lead exposure during a defined growth period and the concentration of lead bioaccumulated in the roots and shoots of the cilantro plant. So basically, I wanted to find out, if I water the plant with more lead, is it going to absorb more lead from the soil? And so I will talk about that more at another time. But
In Sierra's defense, she actually has some really cool data showing that I'm not going to tell you what, but she is doing an EOG later this semester, so we said, don't show your data.
I will show my data, though. That one, no, that didn't, stop. I didn't mean it like that. Okay, settle down, please. Thank you. Okay, this semester, I guess I should introduce myself. Hi, I'm Claire. I'm a junior biology research major here at Southern, and this semester I did some research with Dr. Trott. We wanted to figure out the concentration of berberine in various commercially available brands of golden seal. Now, we talked about golden seal a little bit, but I'll talk about it again. Golden seal is one of the most popular herbal supplements in the US. It's used to treat things ranging from like a common cold to intestinal issues. And the active ingredient in golden seal is berberine. So these were the 3 brands that we, how do you do that? Oh, Nature's Way, Now, and Pure Mountain Botanicals. And so to do that, we used high performance liquid chromatography. I don't know if any of you guys have taken biochem yet, but you guys will definitely talk about that in biochem. And how that kind of works is we take like an organic solution and push it through this little column full of carbon beads at a very high pressure, and then different pieces in that solution will hang on to those carbon beads and then elute at a certain time depending on their affinity to the carbon, and then to detect what they are, we have these absorbance spectra. So there's Dr. Nelson. He was such a great help, but I'll talk about that later. Anyway, Dr. Nelson and I were slaves to the HPLC machine. She is very finicky. She needs very specific, I found this meme online. I had to put it in. Like temperature, pH, all matters. What buffer you use, all matters. So we spent a long time trying to figure out, problems that we faced, as well as how much time it took to put in everything into the machine. Each run takes like 5 or 10 minutes. We did like 5 runs of 3 different samples. It took like the whole of October 31st, Dr. Nelson and I were in there with the HPLC machine. Also, I don't know if this was a play on words or a typo, but I just wanted to point that out. Rather than be like, oh my gosh, her meme has a typo, that's so embarrassing. Anyway, so how did we do this? Methods. First, we had to make a buffer every time we used the machine, and then we would solubilize the herbal supplements and berberine standard, and then we would pump it through the machine, and then we would repeat that. And if we ran into problems, we'd have to fix them and repeat it again. And that's research for you. It takes time. So why did we use the berberine standard? Well, in order to find the concentration of berberine within these unknown herbal supplements, we took berberine standard and we knew what concentration that was at. And then we would inject it into the machine, and then it'd go through and spit out a certain absorbance. And then it would spit out a certain peak area, and then we would plot those against each other to form this linear regression. And then once we put in the unknowns, then when they spit out a certain peak area, we could just use this equation and figure out the concentration for the berberine of the unknowns. So that's why we did the standard curve. And then these were part of the results. One of the questions was, well, how do you even know berberine is in the golden seal herbal supplements that you're running through the machine? So here's HPLC output of the berberine standard. The asterisk shows when the berberine eluted. And then this is like the absorbance spectra of berberine. It has this kind of characteristic 3 hump thing. And so this is one of the brands, Nature's Way, that we put through. If you notice how similar it is, that's berberine, so good for us. Finally, my results, turns out Nature's Way now had the highest concentration of berberine. So I don't know if you guys use herbal supplements, but there you go. Now it's the one I guess you want to buy or want to avoid, I don't know. And part of the process was, is this statistically significant? Because if you notice, these are kind of small numbers. But thankfully, with the help of Dr. Corbett, we plugged it into our commander and did a one-way ANOVA and found there was statistical significance in the different concentrations. These are the people I want to thank. Yeah. I'm sorry.
So one of the things that all of these students have learned is that there's really neat questions that we can ask by doing research with plants. Of course, plant guy, right? So we got to do plants. But of course, they've also all struggled, and Devin, showed some of the struggles that he's been having with that. Claire talked about, just repeat and repeat and repeat. And one of the things that we learned through research is that that's all part of the process, right? Research isn't about, I'm going to ask a question, I'm going to do this experiment once, and we get the right answer. So I want to encourage all of you to think about the potential for research in the department. I'm trying to get out of this projector because it's blinding me. Please talk to Dr. Nelson or Dr. Corbett or Dr. Acevedo or Dr. Thornton when he comes back from sabbatical, or Dr. Foster, all the other professors who are doing research. We'd love to have you be a part of these sorts of processes so you guys could be up here in a few years and do this with us. So thank you for coming today, and that's it. Love to answer some questions.